Welcome everybody who's hopping on to this month's AITC webinar. I'm happy to be here, and thanks to Anne and Peter Abadir and Roemmich and all the rest of the AITC team that I work closely with for the invitation to come present this month. I'll go ahead and get started, and as people continue to hop in, feel free to interact via the Q&A. I won't be able to see, I don't think you'll be able to enter anything in the chat messages, but I should be able to see the Q&A as we go. I'm happy to answer questions as they come in. If there's some more specific questions, I might recommend that I reach out to you offline. So if that's the case, I'll try to put something in the Q&A. I'll link my email address and we can correspond after the webinar today. But yeah, as I said, happy to be here. I'm Roemmich. I'm the director of the Center for Movement Studies at the Kennedy Krieger Institute. I'm also the director of the Rehabilitation Precision Medicine Center of Excellence at Johns Hopkins. And I'm also one of the PIs of the newly formed PRECISE Center, which is an NIH-funded P50 center focused on precision rehabilitation across the lifespan. So there we use a variety of different data sources and technologies to try to make better predictions about individualized trajectories of recovery in a wide variety of different clinical conditions that affect people, again, across the lifespan. As part of that center, we also do a lot of technology development, and we have a resource core. So if any of you are interested in using any of these technologies that I'll be describing today, please be in touch. My email address will be on the final slide, but I'm also happy to share it in the Q&A if that's helpful. But today's talk will be focused on using artificial intelligence for motor assessment in aging and frailty. And so whenever I start to talk about this work, I always try to motivate it by first sharing the origin story of kind of where this came from. And so my background is in engineering and movement science and biomechanics. Following my undergraduate in engineering, I did a PhD working largely with a movement disorders center at the University of Florida. And then ever since then, I've been at Hopkins and Kennedy Krieger working first as a postdoc in the Department of Neuroscience in a rehab-focused lab. And then I've been on faculty here in the Department of Physical Medicine Rehab at Hopkins and also at the Kennedy Krieger Institute since 2017. So all my work focuses really on how we can understand how people move and how we can help them to walk better. So at the core of this talk today is really going to be this question. So how should we measure human movement? And in the past, the ways that we've done that are by either using what you see here, a big three-dimensional motion capture lab. Some of you may be familiar with these technologies. So this is, as I was describing, a three-dimensional motion capture laboratory. These are the types of spaces where you may have seen things like the behind the scenes of how they make Avatar movies or how they make Toy Story, computer animated characters, where we'll have people come into a laboratory like this wearing physical markers all over their body that are then tracked with these three-dimensional motion capture cameras. So these are high resolution tools and technologies that can provide really granular data about how somebody's moving in a three-dimensional space. Then on kind of the other end of the spectrum, we have things like wearable devices that don't always provide nearly as much information about what we would call movement quality or how well somebody's able to move, but rather these technologies provide a lot of information about movement quantity or how much somebody's moving. Obviously, these can be worn in daily life. They can be used to track physical activity in community settings or in clinical settings. But in the past, you've kind of been at one end of the spectrum or the other. Either you're getting this really high quality granular data about the person's biomechanics, or you're using these more scalable remote monitoring technologies for getting information about how much somebody's moving outside of the laboratory setting. And so my previous work was largely focused in a laboratory like this. As you can see here, this is actually a photo of our lab. We have a similar space that I just showed you with these motion capture cameras around the top of the room. But in 2019, the lab looked like this. And so you can see here, there's not supposed to be this big dryer in the middle of the floor, and the carpet's not supposed to be all torn up all over the lab. We had a big lab flood that put our lab out of commission for several weeks, as you'll see here. And then in about 3 weeks later, we had another flood that extended that downtime for another month plus. And in the middle of this, my first son was born. So this is my son, Sam. And so you can imagine this is not the least stressful point in my life, but it really caused me to start thinking about things like, how do we start to get this high quality movement data without access to these three-dimensional motion capture laboratories where people need to come in person and wear these markers and spend at least an hour in the laboratory being evaluated. And at that time, I also had a new postdoc. This is Dr. Jan Steenum, who is now an assistant professor in the Department of PM&R at Hopkins. And you can see here, Jan arrived right after all this flooding happened and while the lab was still down. And in addition to that, you'll notice that he arrived at a peculiar point in history where he came in October of 2019, and he actually was coming to work on a project focused on measuring the energetics of movement. And so to do that, that research required participants to wear this reusable mask on their face. And so you can imagine about 4 or 5 months after Jan started, people were not so interested in coming into our lab in March 2020 to wear a reusable breathing mask on their face. So because of these two situations, the lab downtime, plus the really inability to do the research that we had planned to do, we really pivoted into a third way of measuring human movement. And that's what I'm going to talk about today. So this third option that I'm going to talk about is called human pose estimation. And so what pose estimation is an artificial intelligence-based technology that uses computer vision to automatically track all these different anatomical landmarks that you can see on this person's body using nothing more than a simple video recording. So in other words, you could take out a smartphone, you could take out a tablet, a laptop webcam, record a video of your patient or research participant, and get back something really informative to inform your clinical decision-making or to provide you with a research outcome that's really granular in nature. You can get really high quality kinematic movement data from your patients or research participants using nothing more than these common household devices. And so today I'll dive into how we've used these technologies, some validation efforts that we've done, and especially how these have been applied in both aging populations and other age-related clinical conditions like stroke and Parkinson's disease. So to give you an example of how these work, here's a simple video of somebody walking across the floor in a gait lab. Nothing particular to notice here other than the person is wearing motion capture markers. Those are not necessarily relevant to these video-based technologies. You don't need to be wearing those markers for these computer vision technologies to work. But as you'll see, once we apply the computer vision technology, the pose estimation tracking, you can see that same person walking across the floor, and now we're able to track all those different anatomical landmarks on a frame-by-frame basis, as I just described on the previous slide. And when we've started to test this, we've also wanted to know, okay, well, a lot of these different algorithms that are doing this automated tracking are really trained using images, these large data sets of images that are primarily images of people without motor impairment. And so for the type of work that we do, it's been really important to start to look at the capabilities and potential of these technologies to be able to track movements in people that may not be represented in those image sets that were used to train the original algorithm. So here, to give an example, here's a gentleman on the right who's had a stroke. I'm showing this particular video because you can see that this particular person is wearing an ankle foot orthotic. They have some bracing on their left leg here. And despite that bracing, when we apply these technologies, we're still able to track that person's movement as he's walking across the street here. So this has been really promising that even though these algorithms were not necessarily trained, for example, to track someone's walking after stroke, they can be applied in these different ways. Because I work at the Kennedy Krieger Institute, I am also involved in a lot of pediatric research. And so again, images of children were not overly well represented in these image sets that were used to train some of these algorithms originally. And so here you can see that despite the fact that this young toddler's body is proportioned quite differently than that of an adult, we can apply these same pose estimation algorithms and track her as she kind of toddles over here toward the camera. While my lab does a lot of gait analysis, we don't do only gait analysis. Here's actually a video of me recorded early in the pandemic. You can see we added a hand model to this video. And so I'm sitting here in my living room recording this with an iPhone. And you can see that we're able to add that hand model and track with a pretty high degree of accuracy the movement of that hand opening and closing. One other common question that I typically get is related to what kinds of devices are needed to actually perform these type of assessments. Do you need the latest and greatest iPhone or the latest and greatest camera to be able to do this? And so to take a look at some of those types of issues, we kind of stacked the deck against ourselves and went back as far as we could to find, for example, here, this is one of the oldest known videos of somebody with Parkinson's disease. This video was actually recorded more than 100 years ago. And you can see that despite the black and white nature of the video, and the relatively poor video quality, we're able to track the tremulous movements in this person, again, using the same exact algorithm that we applied to these other situations. So you don't need the latest and greatest video recording technology to be able to do this. You just need something that will give you a relatively good view of the behavior that you're trying to quantify. I always put this slide toward the front of my talk because as we go on, I want to make it clear that in these particular cases, we're not aiming to use these computer vision technologies necessarily to diagnose patients or make decisions for the clinician, but rather the use case that we focus on is providing the clinician with better data that they can use to inform their own clinical decision making. Or alternatively, if we're working with researchers, provide those researchers with better data that they can use as outcomes of their research. And so we always keep the clinician and the researcher at the center of this process, where we imagine recording these videos in an in-home setting or in a potential in-clinical setting, and then feeding that data back through the clinical team to facilitate things like more granular motor assessments or better opportunities for measurement and delivery of telerehabilitation. And so these are the types of things that I'm going to really focus on today. Again, are these ways of getting better movement data that you can use as research outcomes or to inform clinical decision making. Throughout this work, we've interacted with a wide variety of different both potentially users and also a variety of different clinicians who have expressed interest in informing the development of these technologies. And so through all that work, we have distilled this down to a relatively short number of common requests that we then use as design constraints for what we were trying to build. So probably the most common thing that we hear is clinicians don't want any additional setup or equipment that needs to be integrated into their clinical workflow. And following up on that, they don't want to spend any additional time beyond what they're already allocating for their clinical standards in terms of the procedures and assessments that they're performing. They also want a single accessible device. They don't necessarily want to have to calibrate anything or set up multiple different cameras to be able to record the movement patterns that they're interested in. They want to use the device they already have in their pocket and achieve some easily interpretable results they can then use again to either inform their clinical decision-making or understand their research outcomes. And so in summary, what we've been working on is creating technologies that can use a simple single digital camera or other video recording device in a point-and-click manner that clinicians essentially want to be able to pull their device out of their pocket, record a video of their patient or research participant, and get something clinically meaningful or actionable back. And so that's what I'll describe today is how we've progressed through these types of technologies. I want to acknowledge up front, there's been a lot of people involved in this work around Kennedy Krieger and Hopkins, and we've done this, as I mentioned, in a wide variety of different behaviors and in a wide variety of different clinical populations. We have collaborators throughout Kennedy Krieger and throughout Hopkins, some of whom you'll see are associated with the AITC, Dr. Abadir, Dr. Chalapa. We have collaborators at UCLA here, like Rajuda Wilson, and we have a wide variety of, again, different behaviors that we're tracking in different clinical populations in some published work previously, ranging from young children with and without neurodevelopmental disorders to older adults who are frail or who have Parkinson's disease, who have stroke. Again, a wide variety of different clinical populations represented. This work started with this work in 2020 by Jan Steno that we developed during the pandemic so that people could continue to get high quality gait data using simple video recordings while most of the biomechanics labs throughout the country were on at least temporary pause. In doing this, we were lucky to come across an open data set of two different data sources. One, there was ground truth motion capture data, as you can see here. So this is somebody walking across the lab wearing those three-dimensional motion capture markers that I mentioned earlier. And what was important about this data set is we had simultaneous three-dimensional motion capture data along with these simultaneous videos shot from multiple different perspectives. So this is the same person walking across the floor, but with videos taken from each side of them. So in other words, we had synchronized data that spanned both the three-dimensional motion capture marker-based settings and these just two-dimensional videos that were recorded using these kind of household camcorders that you can see on each side of the image. There's a question in the chat about what computer vision models do you use? Are you using the VLA model? This original work was done using OpenPose. So again, I can play this one more time. After we applied OpenPose, you can see now on the top, we have this video that was recorded, synchronized to the three-dimensional motion capture data on the bottom. And once we apply the OpenPose pose estimation tracking, we can then extract these anatomical key points from the video that can closely align with the key points that we see from the three-dimensional marker-based data. So again, it was very important that this data set had both video and simultaneous motion capture data that we could use so that we can validate any sort of gait analysis approach that we might be able to develop for this video-based application. So this is one question we always get right up front of the talk. Okay, this looks really cool. I can see that somebody's walking across the screen with these colorful markers over them, but how accurate is this technology really with an eye toward, again, clinical and research applications? And so just really quickly, showing some spatiotemporal gait parameter validation data here. First, what I'll show you on this first row to orient you to this figure really quickly. In the first column here, on the y-axis, you're seeing the average step time as measured using our video-based approach. And on the x-axis, you're seeing the average step time from the motion capture data. Again, this was in a sample of about 30 younger adults without any sort of gait impairment. So these are basically control participants. In the middle column, you'll see the results for mean step time from the camera on the opposite side of the room, camera 3. Again, against the step times as measured by motion capture data. And in the far right column here, you'll see the data from the two camera views compared to each other. So this is basically showing how reliable are these technologies in relation to each other from different camera views. And so you can see here that when we're measuring step times, we were able to get very accurate data, essentially regardless of which camera we used, and that the two of those cameras provided very similar data to one another. So this is very exciting to us. Step time, we thought, might be one of the easiest things to measure because you don't need any sort of spatial reference. You can simply identify when the person's feet hit the ground and take the difference between those steps in terms of the timing. But it was a good first pass. Then we did set up a system to be able to measure spatial information. Essentially, as long as you know in our system some kind of ground truth in terms of how far something in the room is from something else in the room along the walking pathway, you can use that data, that reference there to essentially convert pixel space into real space. So how many pixels equals, for example, 1 meter? And so once we had that set up, we were able to start deriving step lengths from these videos. And again, using the same format that I described on the top row up there. It doesn't matter which camera angle we use. It doesn't matter, looking at one camera versus another, we see similar results. We found that we were able to measure step lengths, using these videos, again, at a very high degree of accuracy compared to the three-dimensional motion capture data. So this was also really exciting for us. We're able to get accurate spatial information from how somebody's walking, again, from a simple digital video. We also looked at gait speed, because if you have the spatial information and you have the temporal information, you should be able to calculate how fast somebody's walking. Again, we found that this was the case regardless of which camera was used in comparison to the motion capture, and then also for both cameras relative to each other here on the right. And so this was nice because many, as folks on this call probably know, many different studies focused on mobility use gait speed as one of the primary outcomes. And so here we really established, at least in these early participants that didn't have any motor impairment, that we were able to accurately measure walking speed using just a single digital video. One of the real advantages of this type of approach over something like an instrumented gait mat is that it also provides you kinematic data at the level of the individual joints. If you're walking across a gait mat, you can get the spatiotemporal information that I just showed, things like step time, things like step length, things like walking speed. But you don't get information about how much somebody's hip is flexing or how much somebody's knee is extending during their gait. And so this was another early validation effort at looking at, if we compare the different video angles, camera 1, camera 3, to the motion capture in the black, we can see really accurate kinematic traces at the level of the hip, level of the knee, and the level of the ankle. Again, essentially regardless of which camera is doing the recording and which comparison you're making to motion capture. So this was all a really, again, this was our first validation study. We were really excited to see that we were able to capture all these different spatiotemporal walking parameters and all these different sagittal kinematics across the different joints with a high degree of accuracy using these pose estimation technologies. If anybody's interested in working with this software or any of the other technology that I described today, everything that we have done so far, we make freely available on the internet. So we have a couple of different GitHub repositories that have all of the code that you would need to be able to walk through these different workflows and execute them yourself. We also have a series of PDFs that provide instructions to go along with the code that you'll be installing here so that you know how to run it and you'll know how to interpret it after it's done. We also have, and I'm happy to share these with the group too, we have a couple of YouTube videos, one providing a little more detailed tutorial on how to use some of our software, and the other providing another kind of guide to go alongside that tutorial. And so if those are of interest, I'm more than happy to share those after the talk today. Okay, but as I just showed, all of those early videos that we worked with were people walking with this wide angle view of them. We can get multiple different strides. They're done in this big, large motion capture laboratory setting like you see here. The room is wide enough that we're able to position the camera several meters away from the person and get multiple strides of their gait. But we know that if we're really trying to use these video technologies in a scalable way, the real world looks something more like this. We might be doing these recordings in these long, narrow hallways that you might find in somebody's home, or these narrow spaces in a private clinical facility. You might be wanting to do this in an inpatient hospital room or along a long, narrow hospital corridor. And so our next step was to really work on some new software that would allow for recordings in these more narrow spaces that could also provide you with usable data. And so what we're talking about here is really making this technology more flexible so that you could record somebody from the front, you could record somebody from the side, you can record from multiple different angles and still get something that's accurate and meaningful. I'm going to pause right here. There's one question in the chat. If you do take the 3D motion capture device as the ground truth, did you also calculate the accuracy of the body key points from 2D video? We haven't done that just because it's not apples to apples. All of the key points don't necessarily correspond to exactly the same anatomical location where the marker was placed. But this is something you could do if you did place the markers in accordance with the key points that are included in the post-estimation model. So I hope that answers that question. But with this study, this was the next study that Jan had led. This was done in collaboration with the Restore Center, which was a P2C funded center at Stanford, where we really tried to take our approach that I just showed you and make it possible again to record videos from people moving toward or away from the camera rather than side to side across the camera. And so I have some math on this slide. You don't need to necessarily focus too much on it. I can describe to you the concepts that are involved. In essence, what we were able to do is we found that we used some geometric relationships to understand that if we know how far someone is from the camera at any point in time in the video, and we know their relative size that they appear in terms of the pixels in the video, we can use some geometric relationships that are related to the camera lens to estimate how far they have traveled in real space. So for example, if we knew that someone started 10 meters from the camera that was used to record them, and they looked this big in the videos that we're taking, if they move forward such that they now appear this big in the camera, we can use these geometric relationships to identify this delta D, which is delta distance. How far did they move from the starting point to wherever they're seen now? And so when we did some validation of this, we can see here that, yes, if we use this equation that combines these relationships in a way that we can, again, estimate that distance traveled in real space. This is just an anecdotal example, but you can see here we had somebody walk at different levels down the hallway, and we knew exactly how far they had traveled because we measured how far they'd traveled across the floor. When we use this computational framework, which is seen in red here, we can actually predict with a really high degree of accuracy how far somebody has moved using that size change in terms of how they appear in the video. And so then we went through and did a bunch of validation testing in our lab where, again, we compared the video outcomes to the ground truth motion capture outcomes. These results are going to be formatted a little bit differently than the ones I showed you previously, so we'll walk you through these again one more time. Here in the first column, you're seeing, and this is in the first row here, step times. In the first column, you're seeing the camera view of the person walking away from the camera. So in other words, the camera's behind the person. They're getting smaller and smaller and smaller as they walk away from the camera against the ground truth motion capture data here on the x-axis. In the second column, you can see the step times as estimated from a camera view where the person was walking toward the camera. So now they're starting far away, they're small, and they're getting progressively larger as they come toward the camera, again, against ground truth motion capture data. And then finally, again, the two camera views relative to each other, walking toward the camera on the y-axis, walking away from the camera down here on the x-axis. And again, we were really excited to see that now with this frontal view of the participants, whether they're walking toward or away from the cameras, we were able to get good estimates of their step times compared to our ground truth data. Moving into the spatial domain, we saw the same thing again with step length. So using those geometric relationships I described earlier, we're now able to estimate how far someone's moved as they're walking in the video. If we're able to do that, we can calculate their step lengths. And so we are able to find that, again, the videos show really strong data that's accurate when compared to ground truth motion capture data. And again, finally, looking at walking speed, whether the person's walking away from the camera or toward the camera, we're able to estimate their walking speed with a relatively high degree of accuracy using this video-based approach. Moving on, one of the next questions we always get, especially from our clinical colleagues and collaborators, is, are your technologies validated in patient populations? And so starting with this particular study, this was done in collaboration with Drs. Chalapa and Abadir over at Bayview. This was a study where we collected videos on 69 older adults, 25 with 25 who were pre-frail, 25 who were robust, and I think 19 who were frail. And so those are color-coded here, and on all the data I'll show you on this slide follows the same color scheme. The yellow dots are the frail participants, the orange dots are pre-frail, and the blue are robust, or people that did not have any of the 5 freed frailty phenotype characteristics for diagnostic criteria of frailty. And so here you can see, again, we show strong correlations with walking speed with a few outliers, and I'll talk about what we did with those in just a second. And again, strong correlations with both step length and step time when doing this with these different groups of older adults. But importantly, what we've been doing, and this is preliminary data that we are currently writing up in a manuscript, when we compared across these different subgroups of older adults who are either robust, pre-frail, or frail, we found significant differences in walking speed that corresponded with what you might expect, moving from the fastest walking speeds in the robust adults to the kind of intermediate walking speeds in the pre-frail older adults, and finally the slowest walking speeds in the frail adults. And we find similar trends both in step length and step time. With increasing or kind of more severe frailty status, we find shorter step lengths, and with more severe frailty status, we find increasing step time. So again, these older adults who are frail are walking slower, and that's because both they're taking shorter steps and their steps are taking more time to complete. And so this was our first pass at really starting to use these data in a way that we can perform gait assessments on older adults using only simple digital videos and assessing these across the different frailty subgroups. There's one more. Could the patients just hold a ruler walking in front of the camera to calibrate the system first without estimation? So you would need a way to translate whatever's measured from that ruler into the analysis. And so we had thought about, typically how we do it is we'll have marks on the floor or something like that we know the distance between, or if you're doing this in a clinical setting, we'll set up a space where we know that the clinical teams are going to record the video in the same way every single time. And as long as we have 2 pieces of tape on the floor or something like that, we can just use that, and then that doesn't require any effort from the clinician or the patient who's performing the actual walking trial. I mentioned that there's some outliers you can see here, and we definitely don't like to see this degree of inaccuracy between the video-based approach and the ground truth here that you see on the x-axis. And so because of that, this is Jeff Yang. Jeff is a research assistant in my lab who's done a really great job putting together a wide variety of different software applications to both clean up data and hopefully make this easier for people to use. Jeff created this graphical user interface that you can see here. There's also a preprint posted that provides details about this, and it's freely available on Jeff's GitHub, that allows a user to bring in the video and the pose estimation data into this graphical user interface and actually drag and drop these markers to reposition them. So you'll often see situations where, especially if you're on a treadmill or if you're walking in a room that has poor lighting, or maybe the person in the video's color of shoes closely matches the color of the floor or something like that, you'll get little points in time where these key points or these anatomical landmarks are not tracked particularly accurately. And so what this allows you to do is go in and go frame by frame, or you can just do any individual for whatever you might want to, however you might want to approach this editing. But you can manually drag and drop these different key points to move them in the right locations and save that data so that it overwrites your original data. And Jeff has done this in a couple of our studies now and demonstrated both that it can be done to improve accuracy, and it doesn't take that much time, depending on, of course, how long your video is and how accurate you want the data to be. But I'll show you toward the end of the talk a little bit today, something that Jeff has done to quantify kind of the impact of using this pose editor. But that's just to say that this GUI is freely available. You can download it. You can run pose estimation directly within it. You don't need to download anything separately. And then once you've run the pose estimation within this application, you can drag and drop all the different key points in case you need to edit the data. One more question. Oh, how are the manual measurements calculated? Yeah, so that's another thing. With these data, we didn't have, at time of putting these figures together, as accurate of ground truth data. We did the step times manually by having somebody watch the video and timestamp each time someone had a heel strike. In other words, each time someone took a step. And then we used that step time data in conjunction with the fact that we knew how long the walkway was to estimate walking speed. So if we knew the walkway was about 4 meters long and we knew how long their step times were, we can use those two estimates to get walking speed. And then from that, we can back out the step length data. But that's probably another degree of, that probably induces a little more error here because, again, these are more estimates, whereas this was, we think the step time manual annotations are probably pretty robust, but there's probably a little bit of error in these calculations here that comes from doing this manually rather than using a three-dimensional motion capture system. But that's a great question, so I'm glad that was brought up. Okay, so just finishing up on that data cleanup, moving into more of the patient data. This is data from some older adults with Parkinson's disease, where we looked at both the ability to estimate their gait parameters using a sagittal video, using a frontal video, and then making comparisons between the frontal video and the sagittal video. And so here again, I'm showing you step times on the first row. Importantly, in these participants, we separated because some persons with Parkinson's disease can have motor asymmetry such that their walking can become asymmetric, where the right and the left legs can function differently. And because we wanted to get estimates of essentially whether we could track changes in someone's walking that occur at a within participant or within subject level, we had them both walk at preferred speeds and fast speeds. And so you can see whether they're walking fast or at a more preferred pace. We're able to estimate their step times accurately from both the frontal and the sagittal videos, showing you step lengths and gait speeds. We see more of the same. We're able to get pretty good estimates across all the different recording approaches and all the different gait parameters in these older adults with Parkinson's disease. And then we also wanted to demonstrate the ability to kind of customize these workflows to measure aspects of walking that might be particularly relevant for certain clinical populations. And so if you're familiar with Parkinson's disease can often result in stoop posture during walking. And so for that purpose, we measured trunk inclination from the sagittal videos. And we found that the degree of trunk inclination, or the stooping of the posture, that we were able to extract from the video, again, closely matched the motion capture data. Looking at the joint angles, I'm not going to belabor these. These are very similar to what I showed you previously in Parkinson's disease. Now we were able to validate that our pose estimation-based gait analysis approach also is able to accurately measure hip, knee, and ankle sagittal angles across both sides. And then we also did some additional validation in older adults with stroke. And so we followed a similar approach where we had them walk at both preferred and fast speeds. Here, the colors don't indicate right and left, but rather the orange is the non-paretic side, or the side that was less affected by the stroke, and the blue is the paretic side, or the side that was more affected. And again, looking at sagittal versus motion capture in the first column, frontal versus motion capture in the second column, and frontal versus sagittal in the third column. Across step times, step lengths, and gait speeds, we were able to track all these different gait parameters relatively accurately, again, across both walking speeds, preferred and fast. And then in people with stroke in particular, one aspect of their gait that clinicians and researchers are often focused on is the degree of asymmetry in their walking. And so this first row, this row E, shows step time asymmetry, and then row F shows their step length asymmetry. These asymmetries can be relatively small. And so we were interested in seeing how well we could capture these aspects of their gait that can be really granular. And so one takeaway here is, even though we see some, again, continued significance in our correlations between the motion capture data and the video-based data, certainly we're most comfortable measuring these small asymmetries in the walking of these persons with stroke using our sagittal or side view videos more so than the frontal videos. Our system isn't currently set up in a way where you could take these measurements, but we would understand that they're probably going to be less accurate using a frontal-based approach than versus a sagittal. And so that was an important finding from this study too, is if we really want this granular information about the asymmetry of someone's step times or step lengths, right now anyway, we would recommend using our sagittal approach. There's a question about loneliness, social isolation. I think I'll save that one for the end, but I'll make sure to touch on this. So I'll get to that as soon as I can. These are the sagittal kinematics after stroke. So again, hip, knee, and ankle angles. And one of the really interesting things here, talking to this asymmetry of someone's walking pattern, specifically if you focus at the knee, you can see here on the paretic knee, on average, people got about 40 degrees of peak knee flexion when they are swinging their paretic leg. This is like when your foot is in the air and you're actually progressing that leg forward. The knee only flexes about 40 degrees on that side, whereas it flexes up to around 60 degrees on the non-paretic side. And so we were excited about the opportunity to really capture that asymmetry. Again, we showed that we can do it in the spatiotemporal domain with the step length and step times, but here we can also see it in the kinematics. We can see that that knee flexes quite differently on average in the paretic leg than it does in the non-paretic leg. So again, we were enthusiastic about the ability to be able to capture these joint level kinematics using the videos. Okay, and as I suggested, in rehab, we're often focused on studying patients longitudinally and measuring changes in their walking pattern. Same thing, we have an ongoing collaboration with the AITC to study longitudinal changes in walking patterns of older adults in those three subgroups that I just mentioned previously, the robust, the pre-frail, and the frail adults. Here, I'm going to show you data from the persons with stroke and Parkinson's disease. What I'm showing on all of these plots now is the change in step time across, or from the preferred walking speed condition or gait trial to the fast gait trial. So this is the change in step time as measured from the sagittal video. This is the change in step time as measured from the frontal video. Both of these, again, against ground reaction or ground truth motion capture data. And then finally, again, the two videos versus each other. And so now we're showing that across step times, we're again able to, with a relatively high degree of accuracy, estimate changes that occur in step time across different walking trials, changes in step length that occur across different walking trials, and changes in walking speed that occur against different walking trials. Again, importantly, these are not longitudinal data. This is not following patients over time. This is measuring the changes that occur from that preferred speed trial to the fast speed trial. But we thought that this provides a nice kind of first pass at understanding if we were anticipating that someone's walking speed would increase as a result of rehabilitation, or if we were anticipating that someone's steps might shorten over time due to a progression of Parkinson's disease or even aging, can we measure those changes accurately? And this is kind of an initial look at essentially providing some data to show that yes, we think we can, and we have some studies ongoing right now to validate that in a more direct kind of longitudinal manner. Okay, and finally, I'll touch on here how can we use it to get best results? So if you actually want to start doing this, what should you do to kind of get things up and going? And importantly, how should you be recording your videos? This work was done by Chris and John, who is a visiting medical student in the lab, and again with Jan, who I mentioned is now faculty in PM&R at Hopkins. And what we did here is we had data on older adults with stroke, and we compared the accuracy of our gait analysis approach when people were walking on a treadmill versus when they were walking over ground. Of course, one of the main differences is if someone's on a treadmill, they remain directly in the center of the field of view the entire time, whereas if they're walking over ground, they translate across that video. And so we wanted to compare if you had, for example, access to both a treadmill and an overground walking space in the clinic, how should the videos be recorded to give you the most accurate data? So here, again, these are about 40 people with stroke. We're seeing step-by-step data on top, and we're seeing participant-level data on the bottom. So participant-level data just means all of the steps for each individual person averaged together. And again, whether you look step-by-step or at the participant mean level data, we see really good correlations, really strong accuracy between the video-based approach, the sagittal video-based approach, and the motion capture data. This is all for treadmill data. And so when people are walking on the treadmill, we're also able to capture these step lengths and step times with a high degree of accuracy. We then compared, we then included our overground frontal assessments to, again, approximate if you have a clinical setting where you could do treadmill, or you could do overground from the side, or you could do overground from the front. How should you set those up? Here, when we did direct comparisons, we did find that the accuracy or the error from the overground frontal comparisons tended to be noticeably higher than both the treadmill and sagittal. And this persisted when you looked at step length or step time, and also if you looked at absolute errors in either of those parameters. So the main difference here is that we didn't account for whether, we did account for whether the video-based approach either undershot or overshot the data, and the absolute error is just how far is it off, regardless of whether it's too short, too long, what's that degree of error. And so no matter how you look at it, the takeaway message from this is really that the frontal videos of overground walking, we would anticipate that those results are going to be a little bit less accurate than results from either the treadmill or the sagittal view. So if you have these spaces available in your clinic or your recording setting, we would recommend doing these recordings either from a sagittal view of the treadmill or a sagittal view of overground walking. Here's some sagittal kinematics for both treadmill and overground. You can see that these look relatively similar. One issue, as I mentioned, was that when people are walking on the treadmill, sometimes the pose estimation based key point markers can move off of those anatomical locations and potentially label on something else, an inanimate object or something that's occluding the walking pattern, like a treadmill handrail in the room. Those caused a little bit of inaccuracy in the sagittal ankle angles that we were able to measure on both the paretic and non-paretic sides during treadmill walking. And so again, we used the pose editor that Jeff has recently developed we've since gone back in and cleaned up these data to really reduce those errors. And so that's what you're seeing here. When the data are unedited on the left versus edited on the right, this is the mean absolute error of the right ankle angle, mean absolute error of the left ankle angle. You can see there's a handful of trials or steps that had really high ankle errors, probably from some of this inanimate object tracking or mislabeled pose estimation key points. But once those are edited, we can really remove all of those very kind of erroneous data points and fix them up so that they are in with a relatively small degree of error relative to the three-dimensional motion capture data. Okay, the last thing I'll chat about today, moving away from walking, because I assume some folks here might also be interested in measurements at the upper extremity. This is Hannah Kornman. Hannah was another visiting medical student who worked with us on this project along with Jan. And here we brought in, again, some upper limb models. We selected a series of behaviors that are part of the Unified Parkinson's Disease Assessment motor section, which is the clinical standard for manual in-person motor assessment of motor symptoms in persons with Parkinson's disease. And so we did a variety of different repetitive movement tasks. What I'll show you today are some upper limb tasks that we performed. We had people do a hand open and close task. So just take your whole hand and open and close it as fast as you can. We also had them, and these were just, this was preliminary testing in young adults without motor impairment. This was also done during the pandemic. So all of this was done remotely. And we had people match the frequency of their movements to a metronome. So they move very slowly at 1 hertz all the way up to more quickly at 4 hertz. And regardless of how fast the metronome was pacing this participant's movement, we found that the hand closures, which would identify each event, and we can calculate frequency from these events, matched closely to the ground truth events across all the different frequencies. So whether people were moving really fast over here or really slow over here, we can still see really close alignment between the events and the frequencies as identified by the video-based approach versus a manual event time. And these events were manually annotated. Hannah actually went through each of these videos on her own and time-stamped each time like somebody's hand would close. So that's why we have so many ground truth data points here. Hannah did a great job going through and doing all of that. Similar here, we did finger tapping. So this differed from the previous one because your whole hand isn't tapping. You're just using one finger to tap at these different frequencies. And again, looking at these data, we saw really close agreement between the video-based calculations of these parameters versus the ground truth. Okay, finally, I'll touch a little bit too on how some efforts we were working on to make this more accessible and more scalable. So really, we want people to be able to use it regardless of where you're located or what type of technology you have access to. And so one thing that Jeff has also put together is, given the interest in just generally comparisons of different videos, whether these are longitudinal, whether these are in response to a medication, whether these are recording even at 2 different points within the same day. He's created another application here that you can upload multiple videos in and get a comparison of the gait parameters that are tracked between these 2 different videos. And what's neat about this too is that there's also, in addition to the AI that provides the computer vision and pose estimation tracking of the people in the videos, there's a large language model integrated into this software such that once you have these key points, the key point outcomes can be passed into the LLM. And what that provides is an opportunity to customize the type of data that you get out of this system. For example, we considered, if you're working with somebody who's recovering from an ACL injury, you want different information about their walking than if you're working with someone, for example, who's had a stroke. And so you can provide that kind of context into the large language model here. You can ask it, what's the difference in this person's step length asymmetry from the video on the left to the video on the right? And then as you start to ask these questions, and you'll get answers from this large language model, there's an AI summary that you see here at the bottom that will start to log some of the results that come out of that interaction with the LLM. And so we're really working towards something that you could provide that would allow for direct either research or clinical interaction in a way that's customizable for whatever that user really wants to get out of using this technology. So again, whether it's longitudinal comparison or pre-post or again, response, whatever it might be, providing some way to customize this so that each user can get exactly what they want is kind of where we're headed. To be able to scale this, we've also wanted to acknowledge that we have an ongoing collaboration with industry. This is Evan Haas. He's the founder and CEO of a company called Curvature, based here in Baltimore. That is a startup company that has also received some AITC funding to be able to really integrate the technologies that I've shown you into a user-friendly application that can also provide things like patient-reported outcomes, for example, as kind of an all-in-one stop for both patient-reported and more objective quantitative ways of measuring movement. And so we continue to develop new technologies and work with them to get our stuff that we're developing in the lab into hopefully the hands of clinicians and researchers who are going to use this at scale. With that, we have about 10 minutes left, so I'm happy to answer. There's a couple more questions here in the chat that I'll get back to. Just a quick thanks to, again, everyone in my lab who's worked with this, everybody at the AITC who has provided support for all of this work over the past several years. This work is ongoing. We're going to continue to develop new technologies and try them out in new applications with the work that we're doing, again, with the Precise Center, which is focused on precision rehab. And if anybody, there's my email right here, feel free to get in contact with me. I'll leave this slide up as we take any questions in the chat so that you'll have my email address, but I'd be happy to connect with anybody after the talk today too. Okay, going back into the Q&A, there's growing evidence that loneliness and social isolation are independent predictors of frailty progression, not just outcomes of it. Have you looked at loneliness as an upstream signal in your frailty research, and are you collecting multimodal data like voice or conversational patterns? Might provide additional signal helpful to clinicians, something you might want to discuss. Yeah, that's a great idea. We have not collected anything like voice or conversational patterns. We have a collaborator, Steve Zeiler, who is doing something like that in ALS patients, where he has eye movement data, upper limb data, and voice data. And so that certainly seems like something that would be interesting in terms of providing some multimodal data that could help with this classification. I agree. What are the spatial and angular resolutions and accuracy? Can you quantify? Yeah, so if you go look at the papers, we provide all of the error data in addition to just the correlation data. So in those papers, there will be measures of accuracy across all the different either kinematics or spatiotemporal gait parameters that I showed you. So I would just refer you to some of the references that I included here, and I'm also happy to send those out after the talk if they'd be helpful. How short does the distance need for a patient walk? That's a great question. So we haven't, the shortest we've done is about a 4-meter walkway. we haven't done much shorter where you just have like one or two steps. So that's what I can say there is we've gone up to gone down to 4 meters. And with that, it still works pretty well. But I can't provide any more detail on something shorter than that distance. Do you foresee using this in remote care applications? In other words, with OTs or veterans in rural communities? Yeah, absolutely. So I did mention this, but part of what motivates a lot of my work is that I'm from a town of about 8,000 people in rural Nebraska. And so anytime we're building something like this, I always just think about how could this be useful there? Who would actually be using this, and what would need to be done with this technology to make it usable in that setting? So we are certainly interested in remote community or rural communities where this would need to be done remotely. And that's part of that collaboration with Curvature, is really trying to get this in something that can be directly in the hands, probably starting with clinicians and researchers, eventually in the hands of patients too. Do I have a publication on the hand movement sites? Yes, I can put that one in the chat here quickly. Let me just see if I can grab this off PubMed really quick. And I'll start to answer some of these others as I'm searching for that. From Rama, how well do these methods work for unconstrained open source videos as envisaged in moves? These particular ones, the answer is I don't know. I think we'll have to see. We haven't done anything yet that we haven't constrained to be pretty planar with in terms of the camera recording of the direction of movement, but we would have to see how well something like this could be used using just a kind of video recorded from virtually any perspective. So I'm not sure exactly on the answer to that, Rama. As a caregiver for Parkinson's patient, this tech would be very useful for researching the terrible dyskinesia chart. Yeah, exactly. So we our R21 that you see here is actually focused on developing something pretty much in line with what you're describing, a video-based motor assessment set of software tools for measuring bradykinesia and tremor and dyskinesias in people with Parkinson's disease. I did my PhD work, as I mentioned, in a movement disorders center where we focused on people with essential tremor with Parkinson's. So that continues to motivate a lot of the work that we're doing. Let me grab the link to this paper. Here's the DOI for that hand paper that was asked about earlier. Hopefully that will work. Could this technology be used to capture sit-to-stand or turning behavior? Let me share this technology, no, but let me, my colleagues at Stanford that I mentioned earlier with the Restore Center have a web app that you can go to. I will link it right here. And hopefully that will answer that question. How difficult would this be to transfer the software to a portable device that could monitor people in their homes? We found that continuous monitoring is very important. Yeah, so there's some work done using actually Wi-Fi signals that can be transmitted. I don't do that work. That's really meant for more like in-home continuous monitoring. For continuous monitoring, I think we still, with our team, the Precise Center here still does some remote continuous monitoring. We tend to rely on wearables for that more than video. We don't record continuous video of anybody in their home or anything like that. We just have these little discrete short videos that were recorded for the specific purposes of things like walking analysis. But there are starting to be these kind of computer vision based technologies capable of continuous and remote monitoring, both kind of in the clinic and in the in-home settings. Do clinicians need to pay to use your new app for Curvature? Yeah, so Curvature is not my startup. That's just a startup that we have collaborated with. So Evan Haas runs that, and that will be a commercial product. So yes, and they do have plans to apply to FDA for regulatory. They're working on that right now. In your collaboration with the American Heart Association, how would you share how you could get endorsement from AHA to quantify movement? Yeah, so we have we've received several grants from the AHA. We recently received, well, it's just wrapped up actually about 2 years ago, an innovative project award that was focused on developing upper limb assessments for stroke. That data is collected and being kind of analyzed right now, so stay tuned on that. But if there's any questions about our processes with AHA, I have had an AHA Career Development Award and an AHA Innovative Project Award, and I'd be happy to share kind of our processes for submitting those applications and putting them together. If anybody has questions, just shoot me an email. You can see here on my last slide. Okay, it looks like we have about 5 more minutes. I don't see any other questions coming in. And you want me to address anything else, or?
No, that was great, Ryan. I think you got a lot of questions answered, so I appreciate that. And thank you for being with us today. So this will conclude the webinar.
Okay, well, thank you everybody for your attention and for attending. And for those with the AITC, I'd love to collaborate more and kind of see how we can move everything forward together. Thanks, everybody.